Welcome back, students. In this section, I'll be talking to you about the different disorders associated with all the different amino acid metabolisms. Now, usually you might have learned the amino acid metabolism, the important compounds formed from it, and the disorder associated with it. Now, in this, what I have done, I have grouped all the amino acid metabolisms together. I grouped all the important compounds formed from each amino acid together, and now I am talking about all the disorders associated with the different amino acids. So you will have a mixture of different amino acid and what is the disorder associated with that amino acid metabolism. Now, first one is glycine. Naturally, glycine, is there any disorder associated with it? Yes, we have what is called as primary hyperoxaluria. Now, basically this is a protein targeting defect rather than just because of glycine. Now, what is happening is glycine forms glyoxylic acid. Glycine forms glyoxylic acid with the help of alanine, glyoxylate aminotransferase. Now, this aminotransferase is necessary for the formation of glyoxylic acid. Now, this further forms oxalic acid. Now, this enzyme, alanine glyoxylate aminotransferase, should be present in the peroxisomes, whereas in these people in primary hyperoxaluria, instead of being in the peroxisome, this enzyme is shifted to the mitochondria. Minute it gets shifted, there will be a lot of other effects, and what will happen is this oxalic acid will increase, leading to oxalic acidemia and oxalic aciduria. This oxalic acid starts getting excreted into the urine. So if it is getting excreted into the urine, when oxalic acid is present, oxalates, what does the name suggest to you? Nephrolithiasis. So these people are more common to get nephrolithiasis or renal stones. Next, I will talk to you about another amino acid, cystine. Now, cystine is increased in cystinuria. So here I have written cystine, and remember cystine has come from cystine. So cystinuria is where autosomal recessive condition, and this initially one of the diseases included in the Garrod's tetrad. Remember, Garrod was the one who was responsible for the different inborn errors of metabolisms associated with amino acid metabolism, were all due to the initial efforts of Garrod. Now, one of the Garrod's tetrad was cystinuria. Cystinuria is because of transport of amino acids are defective. Now, which amino acid? We give them the acronym COAL, C-O-A-L. In this, C stands for cystine, O stands for ornithine, A stands for arginine, and L for lysine. So the transport of amino acids, this is defective. Where? It could be in the intestine or it could be in the renal tubular cells. Mainly in the renal tubular cells, where reabsorption of these amino acids has to take place. So this will be defective. So what? It will lead to cystine excretion. When cystine is excreted, when it reaches the ureter or the urinary bladder, where the urine is standing for a longer time, it can lead to accumulation of these cystine and result in the formation of calculi. So cystine calculi are seen in these people. There is one more condition associated with cystine, that is cystinosis. Now here there is deposition of cystine crystals in the lysosomes. Where? In the liver, spleen, bone marrow, WBCs, kidneys, everywhere cystine crystals are deposited. Again, this is an abnormality of the transport system. Now, as I was talking of Garrod's tetrad, the four conditions which were originally included in Garrod's tetrad include pentosuria, which is associated with actually the carbohydrate metabolism, Alkaptonuria, the black diaper syndrome, and albinism, and plus cystinuria. These are the four things which initially included in the Garrod's tetrad. We'll go on to methionine. Now, one of the disorders is Osthaus syndrome. Now, here there is malabsorption of methionine and excretion of methionine. Not a very benign condition. So, let us go further. Homocystinuria. Where have you heard homocysteine? Homocysteine is involved in methionine metabolism. So, methionine to homocysteine. Homocysteine can be reconverted back, or homocysteine is converted back to cystine and or homoserine, then to propionyl CoA. So, we know all about the homocysteine metabolism. Now let us look at homocysteine. Nuria, what is that? What are the causes? A lot of causes have been implicated in homocystinuria. Now, most common among them is cystathionine beta synthase deficiency. What is cystathionine beta synthase? Please remember again, we are talking of homocysteine combining with serine to form cystathionine. This enzyme, this cystathionine synthase is also requires pyridoxal phosphate. So absence of vitamin B6 or a deficiency of this enzyme itself, cystathionine beta synthase. Now what else is required for homocysteine metabolism? Either it is to form homoserine and further metabolize to propionyl CoA to succinyl CoA, where it requires vitamin B12, or homocysteine conversion to methionine. Again, homocysteine methyl transferase requires vitamin B12. So either way, cobalamin deficiency will lead to increase in homocysteine. Homocysteine has two pathways and both the pathways require vitamin B12. So no vitamin B12, it will lead to homocystinuria. Another important deficiency is deficient methylene tetrahydrofolate reductase, THFA reductase. Please remember, this THFA reductase is responsible for the conversion of methyl tetrahydrofolate to tetrahydrofolate, or for the real initial formation of methyl tetrahydrofolate, we need methylene has to be converted to methyl. Only then methyl can donate its methyl group. So if methylene is not converted to methyl, the enzyme is deficient, again we can end up getting homocystinuria. Now there are other causes for homocystinuria. Those are called as the acquired hyperhomocysteinurias or hyperhomocysteinemias. Remember, anemia means in the blood, nuria means in the urine. So nutritional deficiency of vitamin B12, B6, folic acid, all these are involved in homocysteine metabolism. Chronic renal diseases are also involved in homocystinemia or homocystinuria. Hypothyroidism is also been implicated in homocystinuria, maybe because of some enzymes are being regulated by the thyroid hormones. That may be the reason why this is happening in hypothyroidism. Drug induced. Which are the drugs? Folate antagonists, B12 antagonists, B6 antagonists, any of these nutritional antagonists, nutritional factors that are required, their antagonists, the drugs can also lead to homocystinuria. So what will happen if somebody is having homocystinuria? One classic feature is these people are more prone for coronary artery diseases. Screening for homocystinemia and homocystinuria is a recommendation nowadays, and it has been advised that whoever is having homocystinemia, they will be given vitamin B6 supplementation to decrease the risk for coronary artery disease. So how does homocysteine lead to coronary artery disease? So before that, let us look at the other features of homocystinuria. Most of these, whatever be the cause of homocystinuria, they will have mental retardation, especially not the acquired, the genetic causes where there is cystathionine beta synthase deficiency. It is characterized by mental retardation, Charlie Chaplin gait, what we call as Charlie Chaplin gait, ectopia lentis, that is the lens are easily removable or you can move around, ectopia lentis, myopia, glaucoma, again eyes features, thrombosis, and in homocystinuria the cyanide nitroprusside test will be positive. When we add nitroprusside or cyanide nitroprusside to the urine, it will immediately form a pink color. So this is a test will be positive. These are the features of homocystinuria. Now what is the relationship between, as I was telling, between homocysteine and coronary heart disease? Homocysteine interferes with the collagen cross-linking by interacting with the lysine residues. Remember, lysine has to be converted to L-lysine by lysine oxidase, and if something interferes with this, there will be no collagen cross-linking. So the collagen will become loose. Now what will, where is collagen? Maybe in maybe in the below the endothelial cells, in the lining the blood vessels. So in this case, it will help in the entry of, it makes the blood vessels bit fragile sort of thing. The basement membrane will not be so strong. At the same time, we have the macrophages and all coming out over there. So it can lead to atherosclerosis. One is that mechanism. The second second thing, it forms homocysteine thiolactones. Homocysteine thiolactone is a very highly reactive free radical. What does it do? It sulfates, or what do you call it? It thiolates LDL. Minute the LDL has been modified, it will be looked upon as a foreign subject, and the macrophages are going to take it up, and more macrophages are converted to foam cells. So that is the reason homocysteine has been implicated in coronary heart disease. Now let us go on to one more important disease associated with amino acid, that is maple syrup urine disease. I have already told you deficient enzyme is branched in ketoacid dehydrogenase, characterized by convulsions, mental retardation, vomiting, acidosis, coma and death, and urine contains branched chain amino acid and it smells of maple syrup, and that is why the name and the Rothera's test is positive. Now we go on to very important disease, that is phenylketonuria. Why is it? Phenylalanine hydroxylase is deficient, or tetrahydrobiopterin may be deficient, or the conversion tetrahydrobiopterin coming and forming from dihydrobiopterin may be deficient. So most important is phenylalanine hydroxylase enzyme is deficient. So what will be the features of phenylalanine? What will happen if this is deficient? So let us look at certain features. We have phenylalanine getting converted to tyrosine. Now in these people, phenylketonuria, this is not taking place. So what? This will increase the phenylalanine. So what? Alanine will get converted to pyruvate. Remember, alanine transaminase. So phenylalanine will get converted to phenylpyruvate. Phenylpyruvate can be converted to, just as pyruvate is converted to lactate, phenylpyruvate will be converted to phenylpyruvate. Remember also that pyruvate can form acetyl-CoA. So phenylpyruvate can form phenyl acetate. I have written it here. Phenyl acetate increases, phenyl lactate increases. So when all these alternative pathway is there for phenylalanine metabolism, when this enzyme is deficient, they go in the alternative pathway. What will happen? They all get excreted in the urine, and they are totally called as the phenylketones. The phenylketones are excreted in the urine, hence the name Phenylketonuria, because phenylketones are excreted in the urine. That is why urine contains phenylketones. Now let us look at one thing has happened, the block in the enzyme, the increase in the increase in the substrate has happened. Now what will happen to the product? Product is not formed, tyrosine is not formed. So tyrosine level decreases. So tyrosine is involved in the synthesis of catecholamines. Remember, tyrosine to dopa, dopa to dopamine, dopamine to norepinephrine to epinephrine, dopamine, norepinephrine, epinephrine. All three are important neurotransmitters. So there is decreased catecholamines, and this means decreased neurotransmission, and most of the times the CNS is affected, and these kids will be having mental retardation. Please also remember that phenylketonuria is an avoidable mental retardation. That is, if you just withhold phenylalanine from the diet, these people can be alright. So that is, even we have screening procedures happening. Even maternal PKU is also screened, or even the amniotic fluid is screened for phenylketones, and we have mechanisms. This is one of the tests which are included in the neonatal screening program to screen for phenylketonuria. The urine of the newborn is screened for phenylketonuria. I'll tell you how it is screened. Let us look further what will happen first. So the CNS is affected, and it can lead to mental retardation because of lack of catecholamines. Remember, decreased tyrosine. Tyrosine is also required for thyroid hormones. If thyroid hormones are not synthesized, the basal metabolic rate may decrease, the brain activity may decrease, which may again contribute to mental retardation. At the same time, thyroid hormones also lead to hypothyroidism. Remember, body weight gain will be there. So the child will be chubby at the same time. He is mentally retarded, but the child also is gaining weight at the same time. So a chubby child actually. Is it enough? It is a chubby child? No, still more things are happening. Tyrosine is also required for synthesis of melanin. So the melanin synthesis decreases. Tyrosinase enzyme is affected. Tyrosinase is not affected. Melanin decreases. So what? It will be a fair chubby child, but it is mentally retarded, and it is one of the avoidable, something which can be treatable, avoidable causes of mental retardation in children. So phenylalanine Phenylketonuria, characterized by fair, chubby child, they say, and with mentally retardation and urine containing phenylketonuria. So what is the lab diagnosis? Can we go for it? Yes, we have what are classic tests for inborn errors of metabolism. One of them is the Guthrie's test. Guthrie's test. Now Guthrie's test is based on one important bacteria known as Bacillus subtilis. Now this Bacillus subtilis requires phenylalanine for its growth. More the phenylalanine, more is the growth. It is as simple as that. So if the phenylalanine content is more, the bacterial growth is more, and we can come to know if the phenylalanine levels are more or not by doing the Guthrie's test. Also, one more thing is usually 24 to before the start of the protein diet itself, neonatal urine has to be screened by either the Guthrie's test or by the ferric chloride test. Now, ferric chloride is a simple test. Urine plus ferric chloride, you will get a transient blue-green color. If it is present, then it is phenylketonuria due to the excretion of phenylketones in the urine. Now, the main thing they say treatment-wise is to give a diet which is low in phenylalanine. Specialized diets for phenylketonuria are available in Western countries. In India, tapioca is one of the vegetables which is deficient in phenylalanine, and tapioca-rich diet can be thought of. This is a preventable cause, and it can this phenylalanine excluded diet can continue in the first decade of life. But there is a problem with that because phenylalanine is an essential amino acid. So its levels are also equally important, not just tyrosine. So a balance has to be maintained, how much of phenylalanine to be given and how much it should not be given, so that it does not cross and get converted to other compounds or cause damage, CNS damage. Tyrosine becomes essential in phenylketonuria. It is essential since this enzyme is deficient. Phenylalanine also in minimal amounts, but when your phenylalanine is excluded, it is essential to give tyrosine in the diet. Tyrosine becomes essential. Now I go on to one more disease, that is Alkaptonuria. Alkaptonuria is called as the black diaper syndrome. Why? Because the diapers on standing turn black. After some time, the urine in the diaper which has been collected turn black. Why? This is because of the enzyme homocysteine oxidase. Remember the metabolism of tyrosine. Tyrosine is further converted to homocysteinic acid, which is further converted to fumarate and acetoacetate. So in this, there is one enzyme, homocysteinic oxidase. If this is deficient, homocysteinic acid levels increase in the blood. So homocysteinic acid is increases in the blood and finally excreted in urine. When it is present in urine, it becomes dark after some time, after exposure to oxygen, and the compound that is formed is called as alkaptone, and the disease is Alkaptonuria. Okay, now there is one important feature of Alkaptonuria that is known as ochronosis. Ochronosis is due to the deposition of these alkaptone bodies in the intervertebral discs and cartilage of the nose, etc. So the intervertebral discs will have blackening, that is known as ochronosis. They can also develop arthritis when black pigments are deposited over connective tissues. Overall, they may lead a healthy life. It may not be so debilitating as phenylketonuria. Remember, because tyrosine is already formed and tyrosine degradation is what is affected. So tyrosine and its catecholamines, melanin, thyroid hormones, all those will take place because those enzymes are not deficient. Only the degradation of tyrosine is deficient. So these people have a better prognosis when compared to higher above, whether it is phenylketonuria or whether it is any other enzymes. If you look at the lab test, we have got the ferric chloride test will be positive, and this Alkaptonuria also gives Benedict's test positive. Remember that Benedict's test is also one of the neonatal screening tests. I go on to one more condition, that is albinism. Albinism is due to the deficiency of the enzyme tyrosinase. Tyrosinase is a copper containing enzyme. When this enzyme is absent, it will lead to the absence of pigment, melanin, from the hair, skin, eyes. These are the main places where the melanin is present. So we have one feature known as oculocutaneous albinism, in which the iris also do not have, iris also do not have melanin pigment, and it will result in irises maybe become pink or red, and more than that, the light will enter freely into the eye, and these people may develop photophobia, nystagmus. They will have light hair, light skin, everything is light in this. This is a genetic disease, albinism. Other include hypertyrosinemia. Let me just finish it off faster. Tyrosinemia type 1, where fumarate acetate hydrolase, it is characterized by cabbage odor. Symptoms last for less than 6 months, within 6 months, and death is more common in tyrosinemia type 1. Type 2 is tyrosine transaminase, characterized by mental retardation, keratosis of the palmar surface, and corneal lesions. Hartnup's disease, we already know, is due to tryptophan metabolism, where absorption of amino acids from the intestine and the kidneys is defective. They have lot of neuropsychiatric symptoms. It has also been implicated that these in certain emperors are supposed to be having Hartnup's disease. Basically, there will be aminoaciduria and pellagra-like symptoms, because tryptophan is necessary for the formation of NAD. Histidinemia is due to the enzyme deficiency is histidase and extragation of imidazole pyruvic acid. This is also characterized by mental retardation and delayed speech development. So with this, we come to the end of different disorders associated with amino acid metabolism. Out of all this, the most important ones, which are exam point of view also and clinically also, is phenylketonuria. Then you have got Alkaptonuria, and then you have got the albinism. Alkaptonuria is due to homogentic acid oxidase. Albinism is due to tyrosinase. Then you have phenylketonuria due to phenylalanine hydroxylase. So we come to the end of all the disorders associated with amino acid metabolism. Thank you.